Hello, my name is Austin Kunz. I am an undergraduate researcher working with the Dr. Lishibanya Moapacho Research Group at the Rochester Institute of Technology, and today I'm presenting my summer 2024 research project, which was analyzing the effects of binding proteins on filaments within a bundle. First, the motivation behind this project. Within cells, there exist cytoskeletons that play a central role in regulating their shape and motility, which often assemble into higher ordered structures called bundles. However, their assembly is not yet well understood. In this project, we modeled a bundle as a collection of filaments held together by binding proteins and simulated their assembly using stochastic simulations. This bundles consist of multiple parallel filaments, shown in blue here, bound together by binding proteins, marked in cyan. These filaments are linear polymers composed of individual monomers or building blocks, which are these free-floating pieces, that are added or removed from their ends at a particular rate. We start with the assembly of a single filament and expand upon that model to analyze how bundles form and the role binding proteins play in that act. This work can be extended to study formation of actin structures like cables and microvilli within cells. Examples of this in real life are figure A here, which are actin cables in budding yeast, which is highlighted in this red line. Microvilli present in our gastrointestinal tract here in figure B, and in figure C, stereocilia, which are present in our inner ear. So first off, the filament assembly, which is the model without binding proteins. This is a single filament with no binding proteins that can either grow or decay. Filaments are biological polymers that consist of monomers that can either grow or decay within a limiting pool of building blocks, and these reactions occur from the end of the filament. So a filament can either grow by adding a monomer to its end or decay by sending a monomer off from its end. The decay rate is kept constant while the growth rate is proportional to how many free monomers are available in the pool. We track this by saying our N is our total number of monomers available in the pool, while L is the length of our filament, so L in this case would be 5. K plus is R prime times L times N minus L, which is our growth rate. R prime is a constant variable, and K minus is equal to D, which is another constant variable, and that represents our decay rate. So this is the theory behind standard filament growth. If a filament has a growth rate that exceeds its decay rate, it will linearly grow as time progresses. And if it has a decay rate that exceeds its growth rate, it will linearly decrease to 0 as time progresses. Neither of these conditions lead to length control. For length control, we would like the rates to balance at a particular point such that the length first increases and then tapers off to a particular size, which is where the growth and decay rate are equal to one another. Thus, length-dependent rates are required for length control. You can see here in this figure, our growth rate is linearly decreasing as time goes on, as more monomers are used up, seen by this formula up here, K plus is equal to 0.5 times N minus L. It's an example of a growth rate. As the number of monomers used goes down, our growth rate goes down in turn, while our decay rate is kept constant at a constant value. And once they reach a point where the growth and decay rate are the same, the length will start tapering off at a length L prime, which is the length at which a filament will have equal growth and decay rates. And this is seen in this figure over here, figure C, which is demonstrating a single filament growing for 100 seconds in a pool with 1,000 monomers, a growth rate of 0.5 times n minus L, and a decay rate of 10. So this is 5,000 trajectories. And as our simulation goes, each filament starts exponentially growing up for the first few seconds, and then as we near 1,000, they start tapering off into a linear, not a linear, sorry, a near slopeless pattern, or just purely horizontal pattern. And there are very limited variations between the filament as it reaches a steady state. See this cyan top line here? It increases rapidly and then smooths off before reaching 1,000, and then it has very limited fluctuations in its size as it continues going. Next, the bundle assembly without binding proteins. So bundles are a collection of filaments that can grow and decay by adding and losing monomers, respectively. They can grow and decay from any end along the bundle. From any filament along the bundle, and they reach a steady state similar to how a single filament would, though each filament tends to be smaller due to sharing the available monomers. So as you see in this schematic here, we have five filaments, which is represented by F. F is 5 for the bindings for the bundle schematic down here. And bundles can grow and decay from any filament. There is no preference based on length. However, there is not a preference based on length. However, we do track the bundle length as the longest filament within the bundle. It's shown here in this schematic here, which is a simulation of a bundle where we have 5 filaments, and we also track the bundle length using the same parameters from the previous slide, except now since we have a bundle, we have to include F, which is going to be equal to 5 since we have 5 filaments. This tracks each of the filament lengths as well as the bundle length, which is a transparent black line on top of the rest. So as you can see, each filament in the bundle is within range of within a steady state range of one another. This top bundle length ends around a length of 240, while this bottom filament 2 is around 170, 175. So they're all within range of one another when they reach the steady state. And the bundle length, once it starts to reach that steady state above 200, it generally tapers off. And there is this end that peaks a small bit, but for the most part, it is very stable once it reaches a steady state. Now, what do the role of binding proteins play in bundle assembly? So binding proteins attach at a point along 2 filaments and act as a glue between those filaments. If one of those filaments is to decay, it and the filament it is connected with will snap at the furthest binding protein location, along with breaking off any connected binding proteins attached to either filament as a result. And therefore, binding proteins serve to both sever filaments and replenish the pool of available monomers. So in these two schematics down here, we have a binding protein that is a binding protein bundle that is set to decay, and the aftermath of that decaying process. So in this first schematic here, schematic A, we can see that one of these we can see that filament 3 here, the center filament, is set to decay. And when a filament is set to decay, it goes to the furthest binding protein along itself and snaps off from that point. So if filament 4 were to decay, it would decay from this binding protein here. But since it's filament 3 that is decaying, it's decaying from this binding protein highlighted in red here. And as a result, this third filament is losing all 5 of these monomers, while the fourth filament is losing these 2 monomers, and the fifth filament is also losing these 3. Now, why is the fifth filament losing those 3 monomers? Because of the binding protein that is glued on between the fourth and fifth filament. Since the fourth filament is going to lose these 2 monomers, and this last monomer of the fourth filament is in effect glued to these 3 monomers here on the end of filament 5, though those monomers will also have to decay if filament 4 is decaying as well, which as a result leads to the new bundle having extremely shortened filaments 3, 4, and 5. Also, we define K attach as A, which is the attachment rate of binding proteins, and A is just a constant. First off, analyzing basic bundle assembly. As a reminder, this is a schematic of a bundle. A bundle consists of multiple filaments, and in our model, we used 5 filaments. Each filament within its bundle has its own length dynamics, which form individual lengths, and the bundle's length is defined as the longest filament. The parameters we use for these simulations are here on the side. We had n equals 1,000 monomers, a K plus growth rate of 0.095 times n minus L, where L is the bundle length. We had K minus defined as just 100, and F, where our number of filaments is 5. So first, in figure A, we have 3 trajectories of a bundle without binding proteins, just showing how bundle length will change in a small sample of time. So each of the bundles starts increasing rapidly in a short period of time. All of them get up to around this 30 to 35 mark, and then experience some shifts and separate from one another. See, one trajectory is a little bit higher compared to the other 2 that are lower, and then they meet up again around this 30-second mark and decay a little bit, and then 2 of them shoot up at the end while one stays a little the same. So these are considered to be in a steady state. There are very little changes as they continue on from the transient state, which is when they're making a lot of changes early on. So they reach a steady state after a bit of time, and that shows in the probability graphs, probability distributions here. So these are in figure B here. These are probability distributions of 5,000 trajectories at various times. So we have t equals 1 in red, t equals 5 in this light blue color, t equals 50 in the green, and t equals 100 in the dark purple. So as we can see in t equals 1, which is when we consider the transient state, the bundle length peaks around 9, and tapers off as it continues on towards 20 in sort of a very thin Gaussian distribution. And as time continues, we go to t equals 5, the slope shifts right, but it also widens. The overall distribution widens to include more values. So that means we're getting closer to our steady state from a transient state. And as we get to 50 and 100, we have curves that are almost the same. The differences can be chalked up to simply simulation differences. And they are much wider normal distributions, if only slightly skewed. But these show that they reach a steady state, and show that they reach a steady state at t equals 50 and t equals 100. Now, the bundle assembly with binding proteins. So the bundle has now been introduced to binding proteins, which bind together the filaments and serve as glue between different filaments. All right, so in figure A here, we have another 3 trajectories of a bundle now with the implementation of binding proteins. So we can see that the slopes, the trajectories follow a very similar path and actually reach a steady state quicker than bundles without binding proteins. However, the binding protein bundles experience much more sharp and erratic cuts in their structure, as seen by these 2 peaks here. We can see that the bundle length goes up, 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 and then just shoots back down. This happens again over here on the second peak. It gets even higher and then shoots back down. And some of these smaller peaks, like this peak over here, this one here, these 2 peaks, and this one, all have these sharp, erratic cuts. Now, this is all due to the binding proteins. So what can be assumed to be happening here is that one filament was getting very long, but then was called on to decay, and it had a binding protein that was very far back. So when it was set to decay, it broke off from that binding protein, which sent the bundle length all the way down because of all the monomers getting removed. We can see similar results in the probability distributions here. Again, these are the parameters for this. We have 1,000 monomers, growth rate of 0.095 times n minus L, decay rate of 100, same filament of 5, and our K attach is just 5, a constant value. So our T equals 1 when we're in the transient state is, again, higher and further, and has a peak that is further left than the rest of the peaks, and it's a little thinner, but it is still a Gaussian distribution. However, T equals 5, T equals 50, and T equals 100, all 3 of them make what is roughly the same curve. They're all smaller than T equals 1 than the transient state, which indicates that they're approaching the steady state. And since T equals 5, T equals 50, and T equals 100 have all have the same form, where they peak around the same value, which is about 8, and extend down to the same value here. You see the blue, green, and dark purple line here all converging at the same point. These all had the same Gaussian distributions as one another, and that would go to suggest that the binding protein bundles reach a steady state much quicker than a bundle without binding proteins would. Although the binding protein state is smaller, like the bundle length is smaller at steady state when compared to the bundle without binding proteins. So we can see that in the comparative probability. So this red and green peaks right here are binding proteins at T equals 50 and T equals 100. And the standard bundle length without binding proteins are here in the blue and dark purple at 50 and 100 as well. And these are just both definitive steady states for each. So you can see that the binding proteins, again, reach a smaller steady state bundle length than the bundles without binding proteins do. However, there's a smaller range of values that the bundle length can be when the binding protein bundles reach a steady state. You can see that this is a very small. This is a very very narrow distribution here. It peaks at around 8 and then continues going down, and the furthest value appears to be at 30. So it's a very thin distribution compared to the bundles without binding proteins, which start at around 3 or 4, go up, peak somewhere in the late teens, and then go down all the way towards the 60s. So it's a much wider distribution, much wider skewed distribution. So which goes to show that while binding proteins reach a steady state quicker, the bundles without binding proteins reach a longer steady state. So as you said, the bundle length will reach a steady state that is larger in bundles without the presence of binding proteins than bundles that contain binding proteins, though it takes longer for bundles without binding proteins to reach that steady state. Binding protein bundles also have more erratic and sharp cuts due to the nature of severing at the point of an attached binding protein, which is seen in these trajectories here. These are these sharp cuts that occur when a binding protein will break off. As for future plans, the main one is to change the rate at which the binding proteins attach to a filament, seeing if increasing or decreasing that K attach value will change how bundles grow, or will change how they decay, and see if that will increase the number of sharp cuts or decrease the number of sharp cuts. I'd like to thank Dr. Lee Shubhanya Mohapatra and Dr. M.D. Sharik Aziz Momin. Thank you to the Rochester Institute of Technology, specifically the School of Physics and Astronomy, and NIH. Thank you.